So now we're going to talk about microbial communities, and then we're going to talk about biogeochemical cycles. I'd rather talk communities first. So, microorganisms, I don't think it's going to come as a surprise. They live in the communities, right? And there are interactions within those communities. You with me? Those interactions can be divided into two broadly defined and not super exact categories. Interactions can be obligate and facultative, obligate and non-obligate, call them whatever you want. So, obligate interactions means, they must, and non-obligate or non-mandatory, they don't have to. So let's start with obligate interactions. The first type of obligate interactions between microorganisms is mutualism. In mutualistic relationships, both partners benefit from the interaction. So for instance, you have two microorganisms, A and B. Microorganism A produces an essential nutrient for B. Microorganism B produces an essential nutrient for A. Okay. Speaking of which, I'm going to define here in the corner the term essential nutrient or growth factor. Very, very simple concept. Essential nutrient, or sometimes called growth factor, it's necessary for growth. It can be anything. It can be amino acid, it can be carbohydrate, it can be nucleotide, it can be vitamin. Sometimes students get confused and they think, oh, when he says, nucleotide, this is an essential nutrient. Anything can be. Does that make sense? So what can a nucleotide be? Yes. Amino acid? Yes. All of that. We good? Does that make sense? Okay. Second type of interactions here is called commensalism. In commensal relationships, commensal benefits, cohabitant doesn't care. Okay, so a pretty common sort of a special case of commensalism is called satellitism. Satellitism is sometimes called nutritional commensalism. I think I have an image, maybe not. Well, I'll illustrate it. So imagine that you have a plate, okay, and you have a streak. I'm going to use different colors. A streak of microorganisms, streak of bacteria that grows in the center. Does that make sense? So this bacteria produce some kind of a nutrient that enables another microorganism grow around it. And only close to it, because when you move farther away, the nutrient is not present. Does that make sense? If you remove the blue streak, the red colonies won't be able to grow, because nothing produces the essential nutrient here. We good? Moving on. Now the third type of interaction, which from clinical standpoint is one of the most crucial ones, is parasitism. Okay. Go ahead.
Commensalism, so the red would be the commensal.
Yes, red, absolutely. Thank you for reminding. So red is commensal. And blue is cohabitant. Okay, now parasitism. Obviously, parasite benefits. At the expense of the host. Okay. Parasitism usually implies that there's a, parasite is inside another microorganism. It can happen. There can be a parasite of a microorganism. They can establish parasitic relationships like this. Does that make sense? Now, in regards to clinical situation, we're going to jumping from microbe interactions to human microbe interactions. In the context of human microbe interactions, parasites can be ecto and endoparasites. Now this is exactly, you should take it at a face value here. So ectoparasites are outside, things like ticks, lice, fleas, Endoparasites are inside, bacteria, fungi, all that kind of stuff. That makes sense? Questions so far? Good? Okay, what about the unobligate relationships? So here, you're going to have synergism, okay, so when A can grow and B can grow, they both can grow, two microorganisms, but when you put them together, they grow much better. Does that make sense? So you see the difference between synergism and mutualism. In synergistic relationships, each microorganism can grow independently. But when they are together, they grow much better. Are we clear about that? Okay, cool. And finally, antagonism, when one microorganism antagonizes another. One cannot grow in the presence of another organism. This will be, for instance, that's just an example, you don't have to memorize it. Penicillium notatum inhibits the growth of Staphylococcus aureus. This is basically how penicillin was discovered by Alexander Fleming in 1924, I think, 27, something like that. Okay, so you notice that fungus, Penicillium notatum, inhibited the growth of Staph aureus. Make sense? This is your source of antibiotics. So far, we clear? Now, you can imagine that microorganisms grow in the communities, but those communities can exist in basically two forms. So the growth can be planktonic, now planktonic growth is what you see in that liquid medium, okay, and planktonic growth is very rare. Most communities exist in the form of a biofilm. And the steps for biofilm formation, there are several of them, they're quite interesting. So it all starts with the colonization of the surface with the primary colonizers. These primary colonizers stick to the surface and start producing extracellular polymeric substances, EPS. What this acronym means actually is in the notes, so you can look it up. It's right there in the notes. But extracellular polymeric substances, they're usually kind of gooey carbohydrates, okay? And they protect, actually, those primary colonizers from desiccation and predation. Now, not only they protect primary colonizers, they facilitate the adhesion of secondary colonizers. Does that make sense? So those are microorganisms that come later. Now you have an established community where microorganisms interact, mostly via mutualistic or synergistic or commensal relationships. Are you following me? Multiply, but at a certain point you can imagine that the space is limited. So these microorganisms will reach so-called critical density. Okay? And at this critical density, microbes start sensing each other. Well, they sensed each other before, but at the certain density of population, the event known as quorum sensing starts happening. So at this critical density, microorganisms produce quorum sensing molecules. Gram-positive microorganisms produce short peptides mostly. Okay. Does that make sense? And gram-negative microorganisms produce various lactones. Now, frankly, I don't really care, like short peptides versus lactones. What I want you to appreciate, gram-positive and gram-negative quorum sensing molecules are different, which kind of makes sense because they have different envelopes, and in order to get them through, they gotta have different structure to cross the envelope. Does that make sense? Now, what are the effects of quorum sensing molecules on the population? All kinds of stuff, like metabolic changes, gene expression changes, even microorganisms may start producing virulence factors at this point. Does that make sense? Good? Okay? Why we care about biofilms? Biofilms are really common in clinical settings. We're talking about like catheters and, prosthetic joints, all that kind of stuff. And microorganisms in biofilms are extremely well protected by EPS. So destruction of biofilms is hard. We should know, people study how they form and how to get rid of them. We're good? Moving on.